therapy for backpack.
Oh, wonderful. Whoa, that's so great.
Yes, and this is Tom Norris. I'm a member of the American Chronic Pain Association. I'm on the, as an advisor for the backpack study also.
Terrific.
And I live with chronic pain.
Okay.
Thank you.
Do we have any orthopedic surgeons attending?
Yes. Well, we have Connor. Maybe you might introduce yourself. He's not a surgeon.
No, I'm not a surgeon, but I do non-operative spine care for back pain at UCSF, and I see one of our UCSF orthopedic surgeon colleagues there.
Yes, this is Mayo Marmo. I'm an orthopedic surgeon. I do some research in pain. Been starting to do it at least. And unfortunately, I will not be able to stay for the whole talk, but I'm very interested in what you have to say.
Okay, good. Well, good. The first part's probably going to be of most interest to the orthopedic group. There's a little bit at the end that I think you'll also find quite interesting.
Will this be recorded in some way?
Jacob? Jake?
It is. Yes, it is being recorded.
It'll be recorded, yep.
Perfect.
Jeff is there too. Jeff is the PI on the backpack study, and he's director, I think, of the orthopedic center or something.
Yes, yeah, thank you. Thanks, Dr. Fields, for presenting. I'm Jeff Lutz. I'm Vice Chair of Research in Orthopedic Surgery and studied back pain for 30 years, approximately. I'm a bioengineer and the PI of the grant, so we appreciate you taking time to educate us today.
Yeah, I can't tell you how happy I am to be part of it, even in a small way.
Okay, let's start.
Okay.
So Irina, I might want to introduce a little.
Oh, yeah. So can you hear me?
Yes.
I can hear you.
Yeah. So it's an honor and pleasure to have you to talk to us and present here. I don't know, Jenny is probably much more qualified to do that, but Dr. Fields is a professor emeritus of neurology and physiology and director of the Wheeler Center for the Neurobiology of Addiction at UCSF. Dr. Fields received his MD, PhD in neuroscience at Stanford and completed internal medicine at Bellevue Hospital in New York. And subsequently, Dr. Fields served in the Army as a research neurologist at the Walter Reed Army Institute, then went to receive clinical training at Boston University of Harvard Medical School, and then ever since he's been at UCSF. So Dr. Fields actually is the one who founded UCSF Pain Management Center that is still functioning really well, and he trained a lot of physicians to continue in his steps. So Dr. Fields made so many discoveries to the pain field and contribution that we can be here all day just talking about it. But I think from my perspective, where I really, that you were a pioneer in bringing the addiction field and pain field together, because before that, we didn't think about that every single substance that produces pain analgesia is also highly addictive. And I think this is where your current work is inspirational and seminal. But among other things that in the laboratory work, Dr. Fields discovered pain modulatory pathway and the role of endogenous opioids and how placebo analgesia works. So I can go on and on, but today's work is going to be something new and something fresh, and that is really amazing. I think it fits so much to the work that we're doing in human pain perception and how this really brain plays a role in susceptibility and vulnerability to pain and chronic pain.
Okay, so can everybody hear me okay?
Yep.
Okay, good. So this is a great opportunity for me to talk to what has become a very large pain community. When I arrived at UCSF, that was in 1972, there really was no specialty of pain management. And it was, most of the pain doctors were in neurosurgery. There was John Adams and Yoshio Hosobuchi, and Ron Miller, who subsequently became the chair of anesthesiology, was doing regional blocks. So we interacted a lot at that point. And then it turned out that there were quite a few patients with pain, and it turned out to be a major clinical need. And of course, as with everything, you know the most when you're starting out. I thought, you know, when I graduated from medical school, I knew everything I needed to know about pain. And what I found out was the more I saw pain patients, the more I realized that I knew very little. I want to start my talk with a disclosure. Let's see. And that is, I'm a neurologist. So I do have a conflict of interest. Obviously, I'm going to try to convince you that low back pain is at least in large part a neurological abnormality or a neurological disease. And what I'm hoping to do is kind of expand your understanding of the neurobiology of pain in a way that can be helpful to patients with low back pain. So there's this canonical model, which has been highly seminal in terms of our understanding of pain. Everybody understands that they're primary afferent nociceptors. These are small diameter fibers. They're highly susceptible to local anesthetics. They're relatively resistant to pressure on the nerve. And this is how it was discovered that the pain signal is transmitted in small diameter fibers. We know that these fibers have their cell bodies in dorsal root ganglia. We know they have their terminals in the superficial dorsal horn. We know that they have a fast transmitter called glutamates. They connect with interneurons in the gray matter of the spinal cord and indirectly to projection neurons, which in medical school you're taught is the spinal thalamic tract. Well, the spinal thalamic tract is, there's a lot of evidence that implicates it. The most convincing is that the fibers run in a bundle in the anterolateral quadrant of the spinal cord, and if you cut it, people lose the ability to feel a pain sensation on the contralateral side below the lesion. In animal studies, the properties of neurons that are in the dorsal horn and project to the thalamus have been extensively studied, and they have properties that are quite consistent with human psychophysics. They show an increased discharge with stimuli of increasing intensity. We also know, and this is actually relatively recent, that the pain signal, when it gets as far as the midbrain and then the thalamus, is then widely distributed in the cortex. There are at least 7 different areas that receive nociceptive information. And that solved one of the neurological problems that have been present since the 19th century, which is that cortical lesions rarely resulted in a long-term loss of pain. And so as recently as, you know, the 1940s, people thought, well, you didn't really need the cortex for pain. You'll see that has really changed quite a bit with functional imaging. We now know that the anterior cingulate and the insular cortex are critical for processing the pain message, and they have activity that correlates with pain intensity. There's also a representation in both the primary and secondary somatosensory cortex. So this is a really great model. You stimulate in the periphery, the message is conducted to terminal regions in the cortex, and with that activity, you have a perceptual event. That's the subjective experience of pain. And this has been, as I said earlier, a very heuristic model. And you can even, such elegant experiments as recording from primary afferent nociceptors in people and in primates and in rats, and showed that the impulses in these primary afferents correlates with subjective pain intensity. Right? So that's pretty amazing. And it's very reproducible from person to person. There's a threshold around 44 degrees centigrade, below which a heat stimulus is felt as warm. Above that, it's felt as painful. And then as you increase intensity, there's a exponential rise in the reported pain intensity. So this is all great. You know, it's a highly elegant, highly reproducible model. And as I said, it's helped us to learn quite a bit about how the pain system works. There's still quite a mystery for low back pain about, you know, precisely which primary afferents are responsible for the pain that these patients complain about. There's no question that there is a peripheral component. That's a major mystery that remains to be solved, and hopefully REACH will solve it. But there are other ways in which this model is incomplete, and I would say to a certain extent has created a barrier to deeper understanding of pain. And I'd like to start off with a bit of clinical information, because ultimately it is the problem that REACH is trying to solve. And this is a study that really struck me as incredibly important. The senior author was Cormier. It was published a few years ago in Pain. And it was a fairly large study. There were over 2,000 patients. There were 3 Canadian multidisciplinary pain centers. And basically what this research did was to assess patients at intake to the pain clinic, and then at period intervals up to 6 months later, right? So this was 6 months studying of 2,000 patients. And the most interesting thing to me that they came up with is that if they asked people whether they thought this multidisciplinary approach, whatever it was, and it was slightly different in each of the different pain centers, if they thought it would be helpful. In other words, they assessed the patient's expectations. And what they found remarkably was that the outcome at 6 months was predicted by the single best predictor of the outcome was not anything that they did in the multidisciplinary pain center, but what the patient's expectation was at intake. So I wanted just to call attention to 4, which is changes in pain intensity, which again is highly significantly correlated with patient's expectation. That's the expected relief. And then pain interference with activities of daily living, which is kind of more relevant to what I'm going to be talking about in the rest of this talk. So here we have a situation where, surprisingly, a psychological expectation on the patient's part predicted what would happen to their pain reports in the periphery. Now, you can't explain that with the psychophysical model. You're going to need some other approach. And this is the subject of my talk. It's a brief outline of what I want to talk about. I've made my first point, which is that the psychophysical model is incomplete. I will give you more evidence on just how incomplete it is. Then I'm going to talk about pain. So we've discussed transmission, which is everybody knows about. Fewer people are familiar, at least comfortably familiar, with the concept of pain modulation. And this area of pain modulation, the neurobiology of it was a major focus of the research in my laboratory and something that I've continued to be interested in throughout my career. Most recently, and what I'm going to talk about within the context of this talk, is a teleological approach. In other words, what is the biological utility of pain? How does the individual use the information about pain to make decisions, right? And I will talk about this in the conflict of different motivations that people are subjected to. In the end, I'm going to get back to the neurobiology and talk about the importance of corticostriatal circuits. And this is something that really hasn't penetrated deeply into the psyche of the pain research community. But in my mind, it's very important if you want to explain chronic pain. You need to know about these corticostriatal circuits because there's really good and accumulating evidence that changes in those circuits are critical factors influencing clinical outcomes of patients with chronic pain. Okay, so first major point, pain perception is not completely determined by the nature of the stimulus. So pain is more than a sensation. It depends on externalities. What do I mean by that? Well, what I mean by that is neutral cues, things in the environment that aren't noxious, are not inherently tissue damaging or potentially tissue damaging, can become factors that modify the pain experience. And the first set of studies that I did, this was with John Keltner, who at the time had just finished his anesthesiology residency at UCSF. And this was done in collaboration with Mark Desposito at UC Berkeley, who had a functional imaging program and was highly collaborative. I really thank him tremendously for the opportunity to do this study. Very simply, what we did was we showed, these were normal, healthy volunteers. We showed them a cue, a visual cue that was red and had high pain written on it. And then we played, a relatively high temperature. Now remember, the pain threshold's around 44 degrees, so 48 degrees is pretty painful. And then we had another cue at a lower but still painful stimulus. This was green with a low pain written on it. That was 47 degrees. So what we did was we consistently preceded and had the cue co-extensive with the stimulus. So the stimulus is applied here, the cue began before and continued throughout the stimulus, right? And then every so often, we would switch and have the high pain cue correlated with the lower stimulus intensity and vice versa, the low pain cue with the higher stimulus intensity. And this shows you the results. So the main point being that if you have, so here, the high temperature with a high cue gave a pain rating of 8.2, with a low cue 7.3. So this is highly significant. So what this is telling you is that these externalities, these environmentally really inherently neutral cues, have a highly significant effect on reported pain intensity. Same with the low threshold stimulus. If it was paired with the high pain cue, that was significantly more painful than the same stimulus with the low threshold cue. Okay, so clearly contextual cues can shape and modify the pain experience. Again, inconsistent with the canonical, psychophysical model for pain. Here's something that is way more impressive. This is a nice study from Fabrizio Benedetti's group in Italy. These, again, were normal volunteers, and they were undergoing a tourniquet pain. So with tourniquet pain, you have a blood pressure cuff on the arm, you pump it up above systolic, and then you have people exercising by squeezing a tennis ball consistently, about 1 per second. And then you just see, well, how long can they do it? And here, they're getting, in yellow, they're getting a saline infusion. So on day 1, they're just getting their pain threshold, their pain tolerance measured. On day 2, they're given an infusion. They're told it's a powerful painkiller, morphine. And on day 3, they're given the infusion without, they're given the stimulus intensity without the infusion. You can see that when people are told they're getting a painkiller, there is a significant but small effect. Now, what's really important is these studies shown here on the right. So in this case, on day 2, they actually did receive morphine infusion, and you can see that morphine way better than placebo. So in case anybody says, well, it hasn't been proven that morphine's better than placebo, well, it certainly is, at least for acute pain in normal volunteers. So they did this for a couple of days, and then they repeated the saline infusion on day 4. So if you compare the saline infusion after the conditioning to the saline infusion before the conditioning, you can see that it's a much more powerful effect. And this is an important point. So this is clearly a placebo. Here's a placebo again, but after conditioning. So clearly learning can enhance the effect of previously neutral sensory cues, right? So, and this is a significant, relatively severe pain threshold, clearly increased and increased greater after conditioning. So another lesson from this is placebos can have variable effectiveness in pain depending on what the patient's previous experience had been. So not only are there these factors of, associated cues, but you can actually enhance the learning so that there's a greater either analgesic or a hyperalgesic effect. I want to talk a little bit about some work that Ben Alter and Irina Strigo did here at UCSF. First, there's this phenomenon of offset analgesia. This is work from Denmark, Arndt Nielsen's laboratory. Here, the point is just to tell you that the direction of change of stimulus intensity in the painful range is a highly important modulator, right? So here, they're using a thermal stimulus to the forearm, and they're raising the temperature slowly up to 48 degrees. And remember, 48 degrees is pretty intense pain, but it's very brief. So they raise it up, and then they maintain it for about 3 seconds or 10 seconds. And then they have the person rating their pain intensity continuously. So here's the stimulus in the dashed line, and here's the pain intensity ratings. You can see that there's a lag between the rise of temperature and the reported pain intensity. So here, the temperature is held at 48 degrees. In this case, down below, T1 is 48 degrees, and then there's a short period of raised temperature and then lowered back to 48 degrees. So at this point, we're comparing a stimulus of 48 degrees, but under 2 different circumstances. Circumstance 1, 48 degrees held constant. Lowercase, 48 degrees, but arrived at from a higher temperature, so that, in fact, while it's still a stimulus that's intense and painful, it's much less painful because it's a lower temperature than previously shown. And here, compare the pain ratings. You can see that there's a highly significant analgesic effect at 48 degrees from the fact that it had been preceded by a higher temperature. And so this was what got Ben Alter and I and Irina interested in this phenomenon. If there's offset analgesia, is there onset hyperalgesia? And the answer to that is clearly yes. So here we're going to let's say 47 degrees, we're approaching it from below, right? So here, the red, the dashed line is 47 degrees held constant, and with the solid line is 47 degrees arrived at from a lower temperature. So here it's 47 degrees, but it's rising. And you can see that the rating, the pain rating is higher when the same temperature is approached from below, right? So the direction of change of a pain signal seems to be very important, as important as the absolute temperature. The primary afferents don't show this, right? So this is something that's going on in the central nervous system. How does this happen? How do you get these modulatory effects on pain due to expectation and due to a situation where the brain is computing the direction of change? So, okay, so we know about the transmission pathway, and we now know that there's also a modulating pathway, a top-down pathway. This runs from various regions of the cortex, including those areas that receive nociceptive input. There's a major projection to the midbrain periaqueductal gray. This periaqueductal gray connects back down to the dorsal horn. Remember, this is where the primary afferents contact the pain transmission cells. And you can see that to a certain extent, you've got this huge nervous system converging onto the first synapse in the central nervous system for the nociceptive message. So what's the point here being that the information that gets to the brain isn't just subject to what the primary afferents do. It's under powerful descending control that's partly activated by learned previously neutral cues, by a computation of the direction of change, and by learning over time things that have been helpful in the past to relieve pain. How does this top-down system relate to things like placebo? Well, a nice study from Christian Buchel's lab in Hamburg by Falk Eipert was a senior author. What they showed was that there's a correlation in human brain in areas that include those that we've talked about. So the hypothalamus, the periaqueductal gray, and the medulla, right? So these are areas that show activity that correlates with pain reduction by placebo. So what's nice about this is it says, look, it isn't just happening in rodents. It isn't just happening in experimental animals. This top-down pathway is engaged to a clinically significant degree in humans as well. They also had a paper showing that this effect on reduction of pain correlates with a change in activity in the dorsal horn. Now, they don't have the resolution to say which neurons it is, but it's consistent with the idea that this modulatory pathway is exerting its powerful influence at the level of the spinal cord. So some other work that involved many, postdocs and junior faculty and graduate students in the lab was the discovery that this top-down pathway exerts bidirectional control. So I feel like this is a very important point. It's worth emphasizing that at several of the relays in this top-down pathway, if you actually study individual neurons, you see that they fall into two functionally critical, distinct groups. One of them, which I call the off cell, generally has spontaneous activity in these lightly anesthetized animals. If you apply a heat stimulus, that's shown here in this black bar, and the animal is lightly anesthetized, and it can withdraw its tail, and its tail flick. So the response is shown here. This is a withdrawal reflex. this cell has to shut off for the animal to be able to withdraw its tail from a noxious stimulus, right? And if you make this cell fire with a variety of ways, either stimulating it or giving it an analgesic, then the tail flick doesn't occur, the cell keeps firing. So that's the off cell. Its activity inhibits pain. And then there's another cell, the on cell, which shows a burst of activity just before the lightly anesthetized rat withdraws its tail, right? And you see these two cell types in the PAG and in the RVM, including RVM cells that project to the superficial layers of the dorsal horn. The cartoon illustrates what's going on here. I think this is a nice, simple way to remember that the control is bidirectional. This would be the primary afferent. So if a noxious stimulus is applied, this drives your transmission cell, let's say your spinothalamic tract cell. Let's say you're firing at 50 hertz here, you're firing at 30 hertz here, and then you get a certain level of pain. Now, if the off cells are active and you have inhibition, for any given input, the transmission cell is going to fire less, right? On the other hand, if the facilitating cell is active, for any given input, this cell will fire more. So the important point of this cartoon is that for any given level of noxious stimulation, or even innocuous stimulation, what's reported to the brain is not just what's coming in from the periphery, it's a combination of what's coming in from the periphery and what the brain is saying it expects, right, to happen. If it expects that the pain's going down, I don't need to respond, your off cell is going to fire more, your pain levels are going to go down. That's basically what we saw in the psychophysical experiments in humans. Okay. So this introduces this term called nociceptive dynamics, proving, I think, that neutral sensation can become pain predictors and that pain predictors facilitate pain. Pain relief predictors inhibit pain, and these modulatory effects occur through this top-down circuit. So you might ask yourself, well, why would you bother to do this? You know, why would you suppress such an important warning signal? After all, we need our pain inputs to protect our tissues from damage. So what's going on here? Why would you do that? Why would you have a circumstance when you're going to amplify the pain signal? Why these distortions? How do they help the animal? Well, in general, I like to quote Theodosius Dobzhansky, who in 1960 came out with this expression, nothing in biology makes sense except in the light of evolution. So, and since I got my PhD in a biology department, I have a biological bias. And so what I'm going to talk about going forward is the biological significance. What does this all mean? Okay, I think we can all accept that nociception is tissue protective. Here's a kid who we now know has a condition called hereditary sensory and autonomic neuropathy type 2. What you can see is their decubiti, fingernails have been chewed so that they're missing digits. And of course, the child has a smile on his face, feeling no pain. From a biological standpoint, nociception protects tissues in two ways. One, immediate motivation, escape, protect the tissue. Number two, it's a teaching signal. You've done something, right? And if the outcome is pain, then you're going to be much less likely to do it in the future. So these are the two major biological functions of pain. You can see that both will be tissue protective, one immediately and one in the future, to prevent it from happening in the first place by avoiding situations which would lead to tissue damage. Okay. Note that these functions of motivation and learning have to do with actions, behaviors, observable behaviors. They have nothing necessarily to do with subjective experience, and yet they have effects on subjective experience. So if you want to know what the function of these processes are, think about actions. The nice thing about that for a program like REACH is that actions are observable, and subjective experience is always subject to interpretation. Okay, this is just to illustrate what I just said. There's a set of neutral cues. You engage in an action. If the result is pain, then the probability of this action in the presence of this cue goes down. If, on the other hand, the action results in relief, then the probability of this action, when this cue comes again, goes up. So that's just the difference between reinforcement, in this case, negative reinforcement, and positive reinforcement. Predictions, this is what we're talking about. Predictions inform decisions. Outcome changes predictions. And expectations, which is what we were talking about with respect to the placebo, are the subjective psychophysical correlate of predictions, right? So expectations, by my definition, are conscious and people are aware of them. Predictions are things that go on preconsciously or unconsciously. Okay. I want to talk a little bit about place conditioning. You have an animal that's placed into an apparatus. You can see it here. This is a rat. There are 3 chambers. The animal's allowed to explore. Then you close the door here. You confine, in our case, the rat to one of the environments. Down below, you can see this little camera to track where the rat is. If you do something that's painful, and then you let the animal run, free in the environment, the animal will avoid a place where it had received an aversive experience, okay? And this involves the cingulate cortex in the rat. Similarly, and this is work that was done in collaboration with the Pareca lab. Tamara King was first author on this. In the presence of pain, if you apply a treatment that causes relief, then the animals will prefer that environment. That's what this shows. So in sham animals, if you put clonidine or omega conotoxin, which is an N-channel blocker and has some analgesic effect in people, these are manipulations that have no hedonic effect in the absence of pain. But if pain is present and you get relief, then you get a place preference when you apply these. So, for example, if animals have received pain relief in a given context, they will tend to hang out there because it's been reinforced. So 2 types of learning. In pain, cues and actions that precede pain are extinguished. Cues and actions that precede pain relief are reinforced. And it's important to point out in this regard that all analgesic manipulations are rewarding, right? So drugs that in the presence of pain give relief will become habit forming in the same way that drugs that are rewarding in the absence of pain, like opiates or cocaine, will become rewarding even if there's no pain. But it's quite similar, and I think it's important to remember. Now, I want to talk a little bit about the biology of these corticospinal pathways. So pain typically occurs in the setting of competing motivations. So normally in a psychophysical lab, there are no competing motivations. All you're doing is measuring, the responses to a noxious stimulus. But in real life, these noxious stimuli, the pain always takes place in the presence of conflicting motivations. I'm going to focus on hunger, but there are obviously a variety of conflicting motivations, including in people in a desire to make money. Okay, so you can imagine that animals in a place conditioning device, it's received a noxious stimulus. In this chamber, but then you put some cheese in the chamber. So now you've established a conflict situation where, oh yeah, it's really problematic for me to go there. However, there's cheese there. And so now you've got a conflict. And what I'm going to try to get across is that in real life, pain is always occurring in the context of conflict. So you have to do a cost benefit computation, right? You've got conflicting goals. You've got to have a decision. How hungry are you? So that's going to depend on whether you've been food deprived. How painful was the situation the last time the trap snapped on you? So you're learning, you're going to do a computation. How risky is it? What's the probability, of getting the cheese? So you're constantly making these computations, and then you're making a decision about what you want to do. And that was illustrated very nicely by this paper from Albert Hertz's lab, where he put rats on a hot plate, never turned it on, but he fed them either their regular lab chow or some chocolate candy. So after about 2 weeks, there were two sets of rats, those that had been fed their regular diet and those that had been fed candy. And guess what? The rats that had been fed candy stayed on that hot plate when it was turned on significantly longer, a couple extra seconds on average. So clearly, when an animal is expecting something good to happen, they're able to tolerate more pain. So this is the basis of a model that I published in Progress in Brain Research, a paper I presented a really long time ago in Sydney. Noxious stimulus occurs in conflicting drugs, and animals doing this cost-benefit computation, for the reward, for the pain. You have to do both, and it makes a decision. And either, if the decision is I'm going to respond and avoid the pain, then you facilitate the pain system so you escape faster. If the decision is, oh no, I'm going to wait for the food, I'm going to wait for the reward, then what happens is you're going to turn your off cell up and you're going to suppress the pain. You're going to be able to tolerate more pain in order to get a desired reward. So where does this happen? It turns out that the system that was discovered by Olds and Miller in the 50s and classified as a reward system, it's a dopaminergic pathway from the midbrain to the nucleus accumbens. If you put an electrode anywhere along this pathway, animals will push a lever to receive electrical stimulation there. So it's been thought of as a reward pathway, but as you'll see, it's a little more complicated than that. The important point is that in humans, you can show that activity in the nucleus accumbens, which is where these dopamine cells terminate, there's a correlation of blood flow with the amount of reward that, in this case, people would receive. So this was done at Stanford in Brian Knudsen's lab, and they were Stanford undergraduates, and they were looking at a roulette wheel, and depending on where the ball dropped, they would either lose money up to $5 or they would win money up to $5. And you can see that on average, there was a correlation between the amount of the reward, the blood flow in the nucleus accumbens, and how happy the individuals felt. So that's a nice study implicating the nucleus accumbens in reward. And a couple, postdoc and a graduate student in the lab, Salim Nicola and Irene Yun, now this is quite a while ago, found a set of neurons in the nucleus accumbens that started to fire immediately when a reward predictive cue came on, and continued to fire until the rat got the reward. And if you either inactivated the ventral tegmental area or you put a dopamine blocker into the nucleus accumbens, these responses went away and the animals no longer responded to the reward predictive cue. This requires an input from the medial prefrontal cortex, because if you inactivate this prelimbic cortex here, the response to the reward predictive cue goes away. And that's shown here. This is before blockade. The red line is after the blockade. Both the behavioral and the neuronal firing goes away when these are blocked. So this is just a summary of that circuit. Prefrontal cortex to nucleus accumbens required for responding to reward predictive cue. And this brings us to the really beautiful study that came out of Jing Wang's lab and shows the power of optogenetics. Now, we know that prefrontal cortex has a variety of places that it projects to. There are a lot of things it responds to, but there are ways of picking out subsets of neurons, and that is through this technique called optogenetics. And what you do is you take a viral construct, which has a gene that is sensitive to light. A gene that encodes a protein that's sensitive to light and opens a pump. And in this case, it's an excitatory conductance. So you can put the virus in the prefrontal cortex. Then this optogenetic protein, this optically sensitive protein is transported down to the nucleus accumbens. In this case, you can shine the light in the nucleus accumbens. The only thing that's going to be activated are cells that have their cell bodies in the prefrontal cortex and project to the nucleus accumbens. So this is a very small percentage of all the neurons that are here, but you can selectively activate them. That's what's shown here. This is where the light probes were. This is where the virus was injected. And here it shows that when you activate the fibers in the nucleus accumbens, you actually get an analgesic effect. So here there's a lot of allodynia, and that goes away when you activate the terminals of prefrontal cortex in the nucleus accumbens. So it's an analgesic effect. And if there's ongoing pain, so for example, with the sciatic nerve injury here, and you shine the light, the animals like it, which means that you've reduced an aversive state. Furthermore, there's an increase in sucrose consumption, right? So not only does this projection from prefrontal cortex nucleus accumbens relieve pain, it promotes reward consumption. So that's a very nice confirmation of the hypothesis that you need to circuit in this particular decision. Nucleus accumbens neurons activated by reward predictive cues. This requires the prefrontal cortex. When you activate this pathway selectively, it promotes reward approach and consumption, and it suppresses pain. Okay, so what about pain predictive cues? Well, it turns out that the nucleus accumbens in humans seems to respond with an increase in activity to pain predictive cues. So this clearly kind of complicates the idea that the accumbens is, a reward encoding nucleus. In fact, we now know that the nucleus accumbens has two sets of neurons, ones that encode what appears to be a reward approach signal, and the other which seems to encode a avoid pain signal. And here, getting back to patients with chronic low back pain, this is a beautiful study. Again, this was done in collaboration with Marwan Baliki and Vanya Abkarian's lab, as published a few years ago. And they took a group of patients with subacute low back pain. So this was low back pain, but was early in the clinical course. So they had about 40 patients that they followed for up to 6 months, right? And then one of the things they found was that there was a loss of gray matter density in both the medial prefrontal cortex and the nucleus accumbens, but functionally, they wanted to correlate activity in these two sites to see if there was evidence of some sort of connectivity between prefrontal cortex and nucleus accumbens, and whether that in any way correlated with clinical outcome. And what we found, was really remarkable. So here, this is subacute back pain. Those who responded to treatment, the R's, in other words, they got better. And then there was subacute back pain that where the pain persisted by 6 months. And you can see that the level of correlation, quote unquote, connectivity between prefrontal cortex and nucleus accumbens was significantly greater in those patients where the back pain persisted. Consistent with the idea that this corticospinal pathway is critical in the clinical outcomes of patients with low back pain. So Neil Schwartz was one of my last postdocs in the lab, and we decided to set up a model with a conflict situation to see what the role of this corticostriatal pathway was. And the behavioral test was quite simple. They received a tone that indicated that a reward was available, and then they could start licking. Then after they learned this task, each one of these vertical lines is a single lick. It's not a nerve cell, it's a lick. Tone, the second tone came on and indicated that there was a 30% chance that there would be a noxious stimulus applied to the rat by laser to the back of the neck, right? So there's a conflict in some of the runs, okay? So either choice 1, they continue to lick. Choice 2, they stop licking when the tone comes on. And clearly, this task was designed just right, because with a small reward, so there are 2 levels of reward. They either got a big reward or a small reward. It just was the amount of sucrose they got. When the pain predictive cue came on, there was a significant reduction in licking. So the animals got less of the reward. Okay, and here is when the reward's no longer available. Right. Interestingly, after about 10 days, most of the animals, started to, go back to licking, right? They were able to ignore the pain predictive cue. In the presence of chronic pain, so these are animals with sciatic nerve injury, then even in trained animals, for the small reward, the pain predictive cue disrupted the feeding. For the large reward, the pain predictive cue did not disrupt the feeding. Okay, so animals learn to feed despite a pain predictive cue, right? In the presence of chronic pain, their ability to do that is reduced. And this just shows that if you make a lesion in a certain part of the prefrontal cortex, in this case, the infralimbic cortex, you can completely abolish the rat's ability to ignore a pain predictive cue. So this implicates at least the prefrontal cortex. Furthermore, if you go back and do this optogenetic experiment where you're injecting the virus in the prefrontal cortex and you're stimulating the terminals in the nucleus accumbens, right? What you find is normally the pain predictive cue is, and this is in the presence of pain, is going to disrupt feeding. On the other hand, when you're stimulating the terminals of this prelimbic input, now the animals continue to feed. So what this clearly establishes is this area in the prefrontal cortex is necessary for the animals to learn to ignore a pain predictive cue. In summary now, this is the original sort of canonical model: noxious stimulus, nociceptive transmission, escape pain. When there's a reward predictive cue, a pathway from medial prefrontal cortex to nucleus accumbens is required for reward approach and simultaneously to inhibit nociceptive transmission. Conversely, when there's a pain predictive cue and you activate, there's a parallel pathway from, it's a different part of the medial prefrontal cortex, to a pain promoter in the nucleus accumbens, which inhibits the reward promoter, right? Now the animal is no longer able to ignore the pain, right? So in summary, the thing to keep in mind is this concept of decision-making and nociceptive dynamics, that the change in pain intensity immediately predicts pain, or it can predict pain relief. If it's going up, it's pain. If it's going down, it's relief. Future teaching signal, it changes the prediction. So you either have a vicious cycle where you have pain, produces pain predictive cues, makes pain worse, and it just gets worse and worse. So one group of chronic pain patients has a vicious cycle where the corticostriatal pathway is contributing to the development of chronic pain. On the other hand, you've got what I call the virtuous cycle, where you get pain relief cues and the reward predictive cue, now the relief becomes a reward predictive cue, and that suppresses pain. So clearly, it's important to deal with patients' expectations. Ongoing nociceptive input produces a bias toward pain-avoiding responses. It shifts the decision-making toward enhancing pain, which is why many chronic pain patients have widespread pain, a variety of pain complaints, not unusual. They have a decreased incentive to engage in rewarding activities, which we sometimes call anhedonia and depression, and probably explains some of the ways that socioeconomic factors influence the reported pain intensity. It's going on in the brain. Clinical implications, very, very important to assess patient expectations in any studies of chronic pain. Patients pessimistic, it's an uphill battle to get them better. So initial treatments are incredibly important. Early successes, early failures have opposing effects. So what you do is very important. The success of it's very important. Very important to keep that in mind if you're in a situation where you want to assess the value of a particular treatment. Helps if you believe in what you're doing, obviously. If you have a good rationale for it, scientifically, that's even better. And if you explain it to your patients, sometimes that in and of itself will be helpful in switching their expectations. And with that, I want to thank my collaborators. I want to thank you all for inviting me to give this talk. And hopefully I'll have some time for questions.
If there are questions, please remember to unmute yourself or raise your hand in the chat dialogue.
The talk couldn't have been that clear. There have to be a few questions.
This is Jeff Lotz. I thank you so much for your presentation. I have a question about then placebo and treatments are really just targeting the placebo effect versus targeting the tissue sources of pain. And as we're thinking about treatments that may be included in some future interventional trials, how important is it for us to be managing the placebo effect as we're assigning patients to more traditional treatments such as physical therapy or others?
Well, obviously I'm going to say it's very important, but I think that one of the ways you can deal with this is, that it goes back to that Cormier study, that you can segregate the patients into different groups depending on what their expectations are. So if you have, let's say you have a group of people who expect things are going to work, you have another group of patients that expect that they won't work. You can compare the treatments within the group where the expectancies are matched, right? So what it could do is reduce sort of the variability that's due to these placebo factors. So people have always said, well, how do you deal with the placebo in clinical trials, especially over the long term? One of the ways that I think you can do it, and I think other people are coming around to the same opinion, is assess their expectations prior to group assignment. Or you can just segregate the patients by expectations and do your statistics that way. And if you do that, if that's part of your experimental plan, then you won't lose, the statistical power, of the group.
And is there an opportunity for changing expectations with education, where patients are educated about what the various treatments options are, and that might have a better alignment with their choice of preferred treatment because now they have an expectation that that treatment is going to be most efficacious?
I think so. Yeah. That's also something that can be explicitly studied, right? Is education, but dealing. For me, clinically, and this is anecdotal. You can ask people if they don't expect a treatment's going to work, why they expect that. And part of the educational process could be, well, look, expectation plays a role in treatment outcome, right? And it's possible, what I always say is, when you study a large number of patients with chronic low back pain, you have to understand, how did those patients get to you? Very often they got to you because all the initial treatments that were done didn't help them, or if they did help them, they got worse despite that, right? So then the challenge is, well, how do you change that person's expectations, right? I think, I don't know how many of you have encountered this, but very often, by the time the patient was referred to me working in a tertiary healthcare setting, they would say, "Doc, I've tried everything and nothing works." And when I heard that, I felt like I wanted to crawl under my desk and say, "Gosh, do you think I'm going to help you?" And sometimes they would say, "Well, no." And then I would say, "Well, but why are you here?" "My wife insisted that I come to see you." And I said, okay, here's what I think is going on. Now, that said, every once in a while, in your examination, you find out something that actually is treatable. One of the things that I did quite a bit of was trigger point injections, myofascial pain. And I don't know how often those dramatic improvements, right, were due to a placebo effect, but for a lot of patients, they did get immediate relief with a local anesthetic injected into a trigger point. And this was one of the few things that I had for patients with low back pain where I could actually get something to happen. Now, what happens immediately is if they get relief, you've changed their expectations. The other thing that we did a lot of in patients with neuropathic pain was schedule them for IV lidocaine infusions. And even if there isn't really an oral medication that you can take to replicate the benefit of blocking voltage-gated sodium channels, just showing an individual that they can get better changes their expectations. So there are things you can do other than simply education. Well, I guess it's part of education, isn't it?
Well, it's an experiential education. So you have
Yes.
to experience that.
Well, those are the most powerful educational effects, aren't they?
Yeah.
By the way, let me just say before I finish that I love what you're doing. I think this is terrific. And I'm really glad that UCSF is part of it.
Yeah, thank you very much. We're excited, particularly because of the very multidisciplinary collaboration and our ability to integrate data from multiple perspectives. And also there's educating ourselves. Some of us have an intrinsic bias. We're biased on the pain generator, nociceptive input side. There's others that are more biased towards some of these modulatory effects. So it's really going to be interesting to see how, if we have strong quantitative data for individual patients, how successful we'll be in using those data to match them to the appropriate treatments.
There's no question in my mind that there are nociceptive generators. We just haven't found out what they are for most patients. And if you can nail those, then you don't have to worry so much about the expectation effects. But until you do, then the expectation remains something that you can really do something to help the patients, even if you don't know what the primary afferent nociceptor is doing or its properties. But I feel like if you guys could get, a preclinical person to actually study muscle and musculoskeletal pain in particular, because 99% of the research that's going on in the pain field is cutaneous pain, which is not a big clinical problem for the most part, except for patients with burn injuries.
Well, thank you very much for your time. This has been a wonderful lecture, and I think we've learned a lot. And we'll have plenty of opportunities to collaborate in the future, so we hope to keep you engaged in our journey.
I got plenty of time, so just call me. And good luck.
Thank you, Howard.
You're welcome.
Thank you.
Thank you.
Bye.
Bye-bye.